Good morning, everybody, and welcome to today's Knowledge Resource Series webinar. We have an exciting presentation ahead of you. We're going to have Jyoti Phatak-Sheldon talking about spatial mapping and confirmation of diverse gene signatures using the multiplexed single-cell approach to the RNAscope assay. So before we go ahead and have Jyoti start, I'd like to give you a little introduction to Jyoti. Jyoti is our associate scientist in the applications team in the genomics division, or ACD division of Bio-Techne. She holds a master's in molecular and cell biology, along with a bachelor's in dental surgery. Jyoti has previously worked in the field of genomics and precision diagnostics as a product specialist for qPCR-based liquid biopsy tests, and is currently an associate scientist in the application teams, executing projects and collaborations that demonstrate novel application of RNAscope and BaseScope technology. And with that, I'd like to hand over the microphone to Jyoti. Welcome.
Thanks, Rhonda. Can you hear me okay?
Yes, we can. Thank you very much.
Excellent. Well, thank you, everybody, to join us this morning or afternoon if you're in the East Coast time to attend this knowledge resource series, with our focus being on single cell studies. And what I really like about single cell studies is that it doesn't really limit us to a certain type of research area. You can really apply this technology across a vast variety of research areas, as long as your research is focused on single cell transcriptomic studies. So I'd like to kind of go over today showing you what the RNAscope assay is, what the technology is, and then show you some examples of publications where researchers have applied this technology in their single cell experiments. So with no further ado, let's jump into it. So what is the RNA in situ hybridization assay? In this whole era of like your discovery tools such as sequencing and PCR, a lot of researchers are now finding the need to validate and confirm their data. And so this technology is really providing you with that ideal spatial analysis tool, really giving you that morphological solution to interrogate these complex tissues that are being studied. Its novelty is that it's highly specific and sensitive to detect these RNA biomarkers in cells and intact tissues while giving you that morphological context, but really showing you that data at a single cell level. And that high specificity and sensitivity is what makes RNAscope stand out in comparison to some of the traditional ISH techniques. Now, this technology consists of 3 parts: the unique target probe that ACD will design against your sequence or gene of interest. Then it consists of the signal amplification system, and this generates a high signal to noise ratio. And lastly, you can visualize your single RNA molecules as dots. The assay will allow you to spatial map various kinds of RNAs, such as long non-coding RNAs, splice variants, and highly homologous sequences, both in cells as well as in intact tissues. And you can visualize your data either under a fluorescent microscope or with a chromogenic label. And it can be widely used across various sample types like FFPE, fresh frozen, fixed frozen, PBMCs, and also cultured cells. And I will show you some examples of these unique sample types in some of the publications later on. So I talk about the assay having a high sensitivity and specificity, and those being our key features. How do we achieve those? Those key features depend on our probe design as well as our signal amplification strategy. The oligonucleotide target-specific probe that ACD designs is depicted here as the Z, and it is to emphasize that those 2 regions that you see are connected via a spacer. Now, the bottom of the Z is what is complementary and will hybridize to your target transcript, while the top of the Z, it creates this sort of a base for that amplification structure to be built. So now when you have these 2 Zs hybridize in tandem to the target sequence, it will create a binding site upon which the preamplifier can be bind and an amplification tree can be built. So after the Z pairs hybridize with the target RNA, the preamplifier binds to the top of the ZZ pair, and to this each preamplifier, you can bind multiple amplifiers. And to each of those amplifiers, you can further bind multiple label probes. As I mentioned before, these label probes can either be a chromogenic label or a fluorophore, depending on the type of assay you want to run. And you can generate a signal which can be detected either under a standard bright field microscope or under a fluorescent microscope. And this particular signal amplification strategy is what yields you that high sensitivity and allows you to visualize your target RNA as a single dot. Now, I also talked about specificity. So how is that achieved? Background is eliminated because this signal is dependent on both of those Z's binding next to each other on your target sequence. And if both those Z's do not bind next to each other, then the preamplifier cannot form a stable hybridization, and the amplification structure does not get built. Consequently, no amplification of any non-specific hybridization also does not occur, which generates almost little to no background signal. Now, a standard RNAscope probe will consist of about 20 of these Z pairs pulled together, and they are designed to hybridize next to each other along your target region. However, you only need a minimum of 3 of those Z pairs to bind next to your target RNA in the target RNA sequence to generate enough signal for molecular detection. And why you may think this is important, especially for researchers that are using FFPE samples, where you know that RNA quality can often be degraded. So this particular strategy really allows you to detect that degraded RNA in those archival FFPE samples. So taken together, the combination of this probe design and the signal amplification tree allows you to give that high signal to noise ratio from your experiment. So I talked about RNAscope technology. That being the technology, we do have 3 unique assays that fall under that technology, allowing you to spatially map or look at gene expression. The first one is the RNAscope assay. The RNAscope assay will consist of about 20 Z pairs per target, and it is usually used for targets that are over 300 nucleotides, such as mRNAs or long non-coding RNAs. Currently, you can do it as a singleplex or duplex chromogenic assay, or you can use fluorescent assay to multiplex up to 12 targets simultaneously. Now, if you're interested in slightly shorter targets that fall between 50 to 300 nucleotides, then you can use the BaseScope assay. The BaseScope assay has anywhere from 1Z to 3Z pairs, and this is usually used for detecting splice variants, highly homologous sequences, or even point mutations. This assay is currently available as a singleplex or a duplex chromogenic assay. And lastly, we recently launched our microRNAscope assay, and this new probe design strategy allows you to really detect those small, short, or really short targets between 70 to 50 nucleotides. So targets such as ASOs, microRNAs, siRNAs, and smaller sequences. And this assay is currently available as a singleplex chromogenic platform. Now, all of these assays, or some of these assays, can be performed either manually, or you can also do them on an automated platform, such as our Leica or Ventana platform. So this is the kind of broad portfolio of the RNAscope, BaseScope, and microRNAscope assay. As I mentioned, the RNAscope has the chromogenic single duplex. You also have the multiplex fluorescent assays that you can do either 3-plex, 4-plex, or even up to 12-plex. Then the BaseScope is a singleplex, duplex, and the microRNAscope is singleplex. But all of these assays do require you to use the ACD probes. Now, currently, we have over 26,000 probes in catalog. But as long as you know your sequence of interest and the target you're interested in is not in our catalog, ACD can design that probe for you for any target or any species in as little as 2 weeks. The only thing is you need to know your sequence. Now, we often have customers get custom sequences or order catalog sequences, but we've seen some customers in the industry, especially in the field of single-cell neuroscience, use some commonly ordered probes, especially for detecting different types of neurons. So we do sell these bundles of ACD probe sets that have some of these commonly used probes in pre-assigned channels. And this is supporting our HiPlex assay that allows for detection of 12 targets. And I'll talk a little bit more about that assay later on in the presentation. Along with all of these product portfolios, we also have this new workflow that customers can now use to kind of look at that sort of multi-omic approach, right? You look at the gene expression or mRNA expression, but also look at protein on the same tissue section. And so with this new co-detection workflow, you can visualize both RNA and protein on a single tissue section. The workflow starts with you permeabilizing your cells or tissue, then binding your primary antibody, then going through a cross-link and hybridization technique for the RNAscope assay and the amplification, and then visualizing the RNA dots and the secondary antibody. So what are some of the applications of this co-detection workflow? You can correlate RNA protein co-expression. You can examine cell type specific gene expression. Some applications that we've seen in the field are that you can also validate neuroantibodies, as there are currently no other visual techniques or tissue-based techniques to do that. You can visualize cellular localization of RNA targets. You can also, given COVID-19 and in the infectious research field, you can detect pathogen at different replication states using also their host cell markers. So really giving you like a broad range of applications. And we've seen a lot of researchers utilizing this workflow more and more with their RNAscope probes. And I will show you some examples of that as well. So with all of that, let's now divulge into RNAscope in single cell studies. Firstly, why is RNAscope important in single cell studies? As we all know, organs such as the brain, the tumor microenvironment, or even studying the different development stages, like all of these consist of these complex and highly heterogeneous tissues. And with this sort of an ushering era of transcriptomic single cell studies, we found a lot of these tissues, they're made up of multiple cell types and states. And some of these cells may be characterized by more than a single gene. And so it is really important to find a particular technique with that high sensitivity and specificity, and a multiplexed spatial approach showing you that data at that single cell resolution, so you can resolve this heterogeneity of these complex cell types. A lot of you in the field in single cell may have, or may be familiar or have seen a slide of the smoothie before, but I always like to kind of relate it to, these organs or these heterogeneous tissues being a form of like an Avengers movies. And doing single cell is kind of diving deeper into each of those Marvel characters, like looking at the Hulk movie on its own or Captain America on its own. And you really get to understand the function and the significance and role of each of those individual cell types within this heterogeneous complex tissue type. So with that, how is RNAscope how does RNAscope fit into that single cell workflow? So as you all know, some of the traditional single cell transcriptomic studies, they use dissociated cells. And it gives you all the data, but it results in a loss of that spatial organization of those cell populations within that tissue context. So that's where this RNAscope in situ hybridization really helps you retain that spatial organization and allows you to confirm and validate your single cell data. It has, of course, a lot of application in various research areas, as I mentioned before. Some of the commonly used applications we've seen in the field are to spatially map a cell atlas. We've seen researchers visualize gene signatures of some of these novel cell types, classify some of highly heterogeneous cell types. In therapeutic industry, we've identified where we've seen customers use RNAscope to confirm some of these new therapeutic cell types. Also in characterizing immune landscapes and identifying some of these new immunotherapy targets. It can also be used to both analyze as well as predict responses to drug treatments. And as we know, there's a lot of publicly available data sets out there, but a lot of times researchers want to confirm some of that data from these publicly available data sets. So we've also seen application in that. So now I'd like to show you some of the examples of how RNAscope is used in the single cell field. Vagal sensory neurons are necessary for control of organ functions, such as the cardiopulmonary or the gastrointestinal systems. But these neurons, they are extremely complex, and the complexity of these and the different types of these neurons, that information is really missing. So researchers from Patrick Armfor's lab at the Karolinska Institute, they presented this comprehensive identification, classification, and validation of the neuronal types in the jugular and the nodus-derived vagal ganglia. So in this particular case, they used about 25 RNAscope probes to not only validate those neuronal types that they identified by single-cell sequencing, but also to quantify the relative proportions of these neurons. So really kind of resolve that heterogeneous cell type population. And so we're showing you some representative data interrogating the nodus vagal neurons. Now, this paper came out before the launch of our RNAscope HiPlex assay that allows for detection of 12 targets. So a case like this is a good example to show that utilizing, multiple tissue samples, if you have a need to look at more than 4 targets, instead of using the RNAscope multiplex assay, one may use the RNAscope HiPlex assay and save some of those precious samples. Now, this paper focuses on the humus epidermis. As you all know, it's a complex organ and it has multiple lineage cells, and it must constantly coordinate with your immune and your nervous system. Now, the epidermal cells of the skin, they have this sort of like a remarkable functional diversity, and that diversity changes and is dependent on its anatomic site. What this translates into is a heterogeneous cell population, and that population, on the other hand, is dependent on transcription of various thousands of genes. So in this paper from UCSF, researcher Cheng and his fellows, they reported a single-cell RNA sequencing of over 92,000 both normal as well as an inflamed human epidermis. And they revealed this sort of a discrete set of specialized keratinocytes that exhibited a distinct composition at different anatomic sites. So really kind of like even within that complexity, there was like a site-dependent variation within those specialized cells. And so this is an application that we've also seen a lot in single-cell studies, where researchers want to compare tissue morphology from a normal tissue to a diseased tissue, really sort of highlighting the significance of a morphological assay. So they used, in that case, the RNAscope multiplex assay to validate their single-cell data by visualizing the gene expression in these scalp keratinocytes and also identifying those subpopulations of the specialized hair follicle cells. So now we've seen examples in neuroscience, we've seen example in skin. Now I'd like to change focus and show an example of single-cell in cystic fibrosis studies. So last year in an issue of Nature, 2 studies profiling the single-cell transcriptome of both mouse as well as human airway epithelium, they both serendipitously identified a new cell type that could be linked to cystic fibrosis. Now, while generating the cell atlases of these mouse and human lungs, these authors identified a new airway epithelial cell type that expresses both FOXI1 and CFTR. A gene, the CFTR gene, as you know, is mutated in cystic fibrosis patients, and it is responsible for transporting the chloride ions out of the cell. This process is normally impaired in cystic fibrosis patients, and that's what leads to the mucus covering the cells. They termed this particular dual positive cell, the FOXI1 and the CFTR positive cell, as pulmonary ionocytes, because they seem to be involved in the transporting of these ions across the lung tissue. Now, the molecular characterizing of this new cell type could aid in identifying an appropriate cellular target for cystic fibrosis gene therapy. And so this is really showing application of RNAscope in single-cell studies for therapeutic purposes. So now this study, talking about those 2 papers, right? This first paper was from Broad and MGH, where the authors, they generated a single-cell atlas of the mouse tracheal epithelium, and they identified functional variations in club cells based on their location. They also identified a distinct cell type in a high turnover squamous epithelial structures, and they named these structures as hillocks. They also identified disease-relevant subsets of tuft and goblet cells. And so they discovered this rare cell type, the FOXI1 positive pulmonary ionocyte, that is also the major source of CFTR in both mouse and human airway epithelium. They also went on to show that the knockout of this FOXI1 in this mouse ionocyte caused a loss of CFTR expression and disrupted airway fluid and mucus physiology, both phenotypes that are characteristics of cystic fibrosis. The other paper that I mentioned about in those studies is this study from Novartis and Harvard, where researchers performed single-cell profiling, and in this case, they did profiling of both human bronchial epithelial cells, as well as mouse tracheal epithelial cells to obtain sort of an unbiased, comprehensive census of cell types in the conducting airway. And unexpectedly, they also identified the same novel rare cell type, those pulmonary ionocytes. So they used RNAscope to validate that both the mouse and the human ionocytes co-express this FOXI1 and the CFTR. And in addition, they demonstrated that CFTR was highly enriched in only the FOXI1 variant, but not the FOXJ1 cells. They also went on to show that notch signaling was necessary and that FOXY1 expression was sufficient to drive the production of these pulmonary ionocytes, that they were a major source of that CFTR activity in the conducting airway epithelium. And all of this data could further inform researchers or therapeutics for cystic fibrosis, really kind of highlighting the role of RNAscope in confirming their single cell data. So now switching gears, this is an application of RNAscope single cell in Alzheimer's disease. Alzheimer's research has a lot of demand in the field, and we've seen a lot of uptake from scientists that have started to sort of use RNAscope and get more spatial data to confirm their findings in case of Alzheimer's. So as you know, Alzheimer's or AD is an age-related neurodegenerative brain disorder. It's extremely molecularly and cellularly complex, and this complexity still remains poorly understood. So MIT researchers in this paper, they analyzed over 80,000 single nucleus transcriptomes from the prefrontal cortex of 40 individuals. And all of these individuals had varying degrees of this Alzheimer's pathology. And so therefore they provided other scientists with this first blueprint for interrogating the molecular underpinnings and the cellular basis of Alzheimer's disease. Now, they actually use the RNAscope duplex chromogenic assay. And this is really helpful because it allowed them to confirm that cell type specific downregulation of these differentially expressed genes, and revealed that significantly fewer excitatory neurons with detectable NTNG1 expression in brain sections from AD patients was seen when compared to patients with no pathology. Once again, kind of going back to that comparison of a normal to a diseased tissue structure and being able to confirm those findings on a tissue context. Now, this is an example of multiple sclerosis, but the reason I included this is because not only our RNAscope assay, but also the BaseScope assay has a good application in the field of single cell, depending on your research. So in multiple sclerosis, as you know, demyelination in the CNS is what leads to the neurodegeneration. But the severity of an MS between patients, or the degree of severity between patients, that is highly variable. And disability does not correlate well with the extent of the demyelination. So that suggested researchers that there were some other factors that contributed to this variability. And this not only holds true for MS, but a lot of other diseases as well. And so in this case, one such factor was possibly this oligodendrocyte heterogeneity, as oligodendrocyte myelinate axons. However, the extent of human oligodendrocyte heterogeneity and its possible contribution to this MS pathology was also poorly understood. So in this paper, researchers from University of Edinburgh, they utilized, again, single nuclei RNA sequencing from the white matter of post-mortem human brains, and they revealed an altered oligodendroglial heterogeneity in multiple sclerosis patient. Now, why this is really important, as you know, with a lot of traditional techniques, post-mortem human brains present a very high challenge of autofluorescence. And our sensitivity and specificity of the assay allows you to eliminate that sort of autofluorescence from these post-mortem human brains and show your signal in a very clear way. So this heterogeneity of these oligodendroglial population that they saw may be important in understanding sort of the disease progression and also developing therapeutic approaches. So as I mentioned before, in this case, they actually used the BaseScope duplex assay, and they complemented the single nucleus data, revealing that correlation between single nucleus RNA sequencing and the BaseScope-ish for subcluster marker expression in the white matter of a human control brain. Now, the BaseScope also showed by segregating pairs of subcluster markers that less than 10% of the oligodendrocytes contained both RNA markers, confirming the presence of these distinct subclusters. Now, in the same paper, the authors also identified through single nucleotide RNA sequencing immune oligodendroglial cells, a population that expressed both canonical oligodendrocyte genes as well as genes associated with microglia. And that's why they named these sort of hybrid genes oligodendroglia. So interestingly, these oligodendroglia cells are enriched in the human multiple sclerosis brain, and this was a finding they confirmed with RNAscope using the dual BaseScope ISH and IHC techniques. So as I mentioned before, the co-detection workflow, they utilized that workflow to look at both the RNA and protein on the same tissue context using the BaseScope probes, as well as performing IHC for the protein marker, really highlighting the significance of being able to utilize RNAscope within this workflow. So with some of those examples in neuroscience and oncology field, I would like to now show an example of single cell application in the field of stem cells. Another area where single cell studies really has a lot of traction and a lot of sort of demand because of the ability to resolve the heterogeneity in these within the stem cell population. So during homeostasis, these LGR5 positive crypt cells, right, they drive sort of a high turnover of intestinal epithelium. But even when these cells are lost, the epithelium still tends to recover. And so they found that there was an additional population of these quiescent stem cells that in the absence of LGR5 was driving epithelial renewal in response to injury. So in this paper, researchers from Alex Gregorius' lab at Mount Sinai Hospital in Toronto, they used single cell RNA sequencing to profile the regenerating mouse intestine and identify this novel stem cell population. And they labeled it as revival stem cells or REV-SCs. Now, what they found was that these stem cells were enriched for genes that were involved in cell survival. And that gene was the clustering gene or CLU. So they saw that during this homeostasis, the CLU expressing cells were rare, but they were distinct from these LGR5 positive cells that are normally responsible for that turnover. And that in response to irradiation, or where they were trying to sort of cause injury, the number of these CLU positive epithelial cells, they expanded. So that was interesting. And so they used the RNAscope multiplex assay, and they revealed that these rare CLU positive cells in the normal intestinal crypt were firstly distinct from the LGR5 positive cells. And that also, again, as I mentioned, that in response to an injury, they saw that there was a significant increase of these CLU positive cells, as you can see in that damaged intestinal crypt. But now, in addition, the researchers identified another gene called YAP1, which was a transcriptional regulator, and it was important for regeneration of intestinal epithelium. Now, they saw that during intestinal regeneration after injury, this YAP1 induced reversal stem cells that will create now the new LGR5 positive cells, thereby restoring the epithelium. And so the single cell RNA sequencing revealed that this YAP1 gene was present exclusively in SITU cluster of those revival stem cells. And they also saw that by deleting this YAP1 gene in the intestinal epithelium, there was a suppressed emergence of these endogenous CLU positive cells. And they demonstrated this again by performing RNAscope ISH along with IF to look at both protein and antibody in the same tissue section. So really kind of highlighting the role of understanding spatial mapping in resolving this complex data that you get from single cell sequencing techniques. So organogenesis, as you know, it requires a complex interaction of multiple cell lineages. And that further coordinates their expansion and differentiation and maturation over time. So this paper is really focusing on application of RNAscope in single cell in development. I talked about before. Development is in another area where there's a lot of heterogeneity within different cell populations. And so in this case, pancreatic organogenesis, it is a complex and a dynamic process. And that process will ultimately result in the generation of multiple cell lineages. And each of these different cell lineages perform the functions of a mature organ, including some very important functions like glucose homeostasis by the endocrine compartment, and also production of digestive enzymes by the exocrine compartment. So in this paper, researchers from Julie Snedden's lab at UCSF, they profiled these cell types within the epithelial and the mesenchymal compartments of the murine pancreas across development using a combination of single-cell RNA sequencing, immunofluorescence, RNA in situ hybridization, as well as a genetic lineage tracing. So really sort of like a multi-platform, a multi-technology experiment to reveal all of the data and all of the complexity of these different cell types. Now, among several major findings, they identified a previously underappreciated cellular heterogeneity of the developing mesenchyme. And as shown here in the E14.5 pancreas, that they confirmed using, again, the dual RNAscope ISH and IF workflow. This particular work, or even other works of the same form, they really establish a roadmap of pancreatic development, and they demonstrate the broad utility of this approach, or this multi-approach, for understanding the lineage dynamics in developing organs. And so this particular example I wanted to show you to kind of highlight that we've seen examples in some FFPE tissues, some fresh and fixed frozen tissues, but really here's an example that RNAscope can be performed across a wide variety of tissues. And this is a paper where researchers have used a whole mouth tissue. So dysregulation of cell type specific gene networks, this results in birth defects. And this can affect about 5% of live births. Now, congenital heart defects, they are the most common malformations, and they result from disruption of cardiac progenitor cells. However, these transcriptional changes in these individual progenitors, they lead to organ level defects that are still unknown. So in this paper, researchers from the Srivastava lab at UCSF, they used single cell RNA sequencing to really interrogate these early cardiac progenitor cells. Once again, comparing them between both normal as well as in abnormal cardiogenesis. And now they performed the RNAscope fluorescent assay on these whole mount embryos to validate all of their major findings and to interrogate all of these cell types that arise during both the normal and abnormal cardiac development of these whole mount tissues. Really sort of highlighting the power of RNAscope in these different difficult cell types that would often present challenges when you use traditional ISH techniques. So I've shown you sort of like a broad area of all applications in different research areas covering, neuroscience, oncology, stem cells, even development. But as we've seen before, a lot of these cell types, they're not often just characterized by individual genes. We have seen more and more cell types now that are characterized by more than just those classic 4 to 5 genes. And so having a technique that allows you to look at all of those targets together and do sort of that higher multiplexing is really important. And that's why I'd like to now kind of give sort of an overview and a highlight on our RNAscope HiPlex assay. The RNAscope HiPlex assay, it provides you signal amplification using that RNAscope technology for up to 12 mRNA targets on a single tissue section. The way it does this is by detecting 4 targets in 3 iterative rounds. And between each of these rounds, you basically go through a fluorophore cleaving procedure. What's really important is that this procedure has minimal to no effect on your RNA and tissue morphology. Once again, a challenge that's faced by a lot of other multiplexing techniques. Using our HiPlex image registration software, the scans from these 3 rounds are combined together, and that really enables you to look at the true 12-plex data visualization. But the key here is, again, you can look at all that at a single cell level with that tissue context. And it is currently compatible with the fresh frozen and fixed frozen sample types. But as I mentioned before, this assay is really, really helpful for researchers that have a need to study, say, more than 4 targets, because it allows you to save your samples by having to run multiple rounds using some of the lower multiplexing assays. So to demonstrate the application of this assay, we utilized this paper by Goksha that was published in Cell Reports in 2016, where researchers at Stanford identified novel subtypes of both the D1 and the D2 striatal medium spiny neurons by analyzing over 1,200 striatal cells using single-cell RNA sequencing. They did perform single-cell RNA sequencing, but at that time, they didn't really confirm or validate their data on morphological context. So they identified these discrete cell types that existed in kind of this continuous spectrum of transcriptional states. And they found that functional diversity within a complex tissue arises from these small numbers of discrete cell types. So we employed the RNAscope HiPlex assay to confirm these results and spatially map these subtypes in the mouse brain. We performed that by using both the HiPlex 8 reagent kit that allows you to look at 8 targets simultaneously, as well as the HiPlex 12 reagent kit to look at 12 targets simultaneously. In the interest of time, I've shown you the data from the HiPlex 12 reagent kit. This is a graphic that was created using an online tool, but it really shows you or highlights the importance of being able to look at tissue-based data, because it really allows you to resolve that heterogeneity of these cell populations, but doing all of that on that same tissue section and not going through that hassle of, repeating these experiments on sort of serial sections. This is a still image from the previous graphic that you look at. And as you can see, we were able to identify both the DRD1 and the DRD2 striatal subpopulations on the same tissue context. Now, you may look at this and you may ask, right, Jyoti, like there's a lot going on. How do I really kind of resolve this data? I can't really see what targets are being expressed where. And that would be a great question. And that's where the RNAscope's image registration software comes into place. Using our image registration software, you can really toggle on and off and kind of play around with your data to understand how these targets are interacting with each other and resolving your data from that heterogeneous tissue. So to show an example, the paper that we used, right, the paper from Stanford, they identified this novel cell type that expressed both the DRD1 and the DRD2 genes. So by just turning on the DRD1 and the DRD2 target, we were able to identify that novel cell type that expressed these two, that expressed both the DRD1 and the DRD2 genes. So you get all the 12-plex data from the 3 different rounds, but it's really the HiPlex registration software that will allow you to kind of play with your data and work, zone into your regions or targets of interest. So I talked about the HiPlex assay. I did want to show you just a couple of early publications that came out using this application, this assay in the field of single-cell studies. So the Habenula complex, as you guys know in the brain, it regulates various behavioral states via the midbrain nuclei. But the transcriptional analysis of this mammalian Habenula is undefined. And so researchers from University of Washington, Seattle, from Garrett Stuber's lab, they performed single-cell sequencing of mouse Habenula complex. And they compared data between the normal mouse and also mouse that was exposed to aversive stimulus. Now, they used the RNAscope HiPlex assay to identify these Habenula clusters. While single gene markers and Cree driver lines have been employed to gain genetic access to Habenula cells, in most cases, the expression of single genes does not accurately reflect Habenula cell types' identities, therefore making the use of HiPlex really, really significant in sort of validating that data. The other example here is out of Cornell. Now, tendons, as you know, they are dense, hypocellular connective tissues that transmit forces between both muscles and bones, right? And so there haven't been many studies that are revealing the cellular heterogeneity of tendons. And tendons are really being studied, researchers are really trying to understand this heterogeneity, especially in studying injury and studying sort of therapeutics for injury. So in this paper, researchers from Cornell and the Hospital for Special Surgery, they performed sequencing to identify 13 unique cell types in tendons. And it was important to understand the complexity of how the tendon extracellular matrix was assembled and maintained. So therefore, you can further understand the designing of therapies to treat conditions such as tendinopathies. Again, really highlighting the importance of HiPlex, where a researcher wants to use more than just 4 genes or multiple targets to characterize some of these novel cell types. So with a lot of these publications, I did want to kind of bring light to this publication. I would like to kind of give a disclaimer. This is still a publication that's on bioRxiv. It's not been peer reviewed, but I wanted to show you this because it really kind of highlights this multi-technical approach that customers have been using. So this was a paper from University of Queensland in Australia, and was a group, a consortium of different organizations. And they presented this sort of a sensitive, high resolution, and genome-wide approach to both discover and validate cell-to-cell communication across a whole spatial landscape of a tumor's tissue section. So they combined the 10x Visium platform, also previously known as spatial transcriptomics, along with single-cell RNA sequencing. And then they also went to perform RNA in situ hybridization using RNAscope and ddPCR to study this cancer in like 2 non-melanoma skin cancers. And they demonstrated that this novel approach to study the cell-to-cell interaction apart from single-cell RNA sequencing with this sort of spatial transcriptomics-based method by refining the target-specific signal using RNAscope assay. So really kind of giving you like that broad overview, using these discovery tools such as single-cell sequencing and 10x Visium products that allow you to kind of look at that bigger picture, right? Or in a way I could say like of a macro analysis, and then really zooming into the image and looking at your targets of interest or those differentially expressed genes and conforming them using the RNAscope assay, as you can see in the lower left image, giving you that single cell spatial context. And also to add on, they try to confirm and sort of add another layer of validation by performing ddPCR on the same sample types. So really kind of highlighting the demand, the need, and the trend of where the industry is going in applying these spatial analysis tools and applying RNAscope in their single cell studies across multiple research areas. So with a lot of these publications that I've shown you, I did want to highlight that the growth and the adoption of this RNAscope technology is really highlighted by the number of peer-reviewed publications that RNAscope uses, And our first publication came out in 2011. And since then, we've had over 2,900 peer-reviewed papers published using RNAscope in various journals. And some of them you'll see in really these top-tier journals like Cell and Nature. But mostly, I wanted to highlight that this technology, we do have some top research areas like cancer and neuroscience and infectious diseases, but you can really apply it across any research area. And that's because of the ability for ACD to design probes for any target or any species, as long as you do know your sequence, right? And even within single cell studies, we see some of these common applications, but there's a lot more publications that I didn't show today in other fields as well. We recently saw a paper utilizing this HiPlex assay for COVID research as well. So really it depends on your nature of the interest of your research and what techniques you're using. But it's important to highlight that morphological data really adds value to all that information that you're seeking or getting out of these macro analysis tools such as sequencing techniques. And with that, I hope I've convinced you all today that the RNAscope uses a unique probe design strategy to dramatically improve your signal to noise ratio. We have currently over 22,000 probes on catalog, but as I mentioned before, newer probes can be designed and made within 2 weeks for basically any target and any species. RNAscope also provides both a quantitative and spatial information on your targets in a complex or a heterogeneous tissue environment, and really giving you that data at that single cell resolution. Our newest assays, BaseScope and microRNAscope, allow you to detect targets such as splice variants and shorter sequences down to either 15 nucleotides or even 17 nucleotides. And lastly, as you've seen constantly across a lot of these publications, that the RNAscope assays can be complemented with both IHC or IF for simultaneous RNA and protein detection on the same slide, something that's really important for researchers that are trying to jump into that multi-omic approach when doing their single cell studies. So with that, I'd like to thank everybody for attending today's webinar. I hope you're all staying safe, but if you have any questions on anything that you learned today, or if you need any more information, you can visit our website, or you can contact our support team, or you could also email me and we'll get back to you as soon as possible. Thanks everybody, and I will hand it back to Rhonda.
Thank you so much, Jyoti. What an excellent presentation. I really enjoyed it. I hope everybody did as well. We had tremendous engagement. We have a lot of questions for you. So while I let you get a little bit ready, I just want to do a plug here for our upcoming knowledge resource webinar series. As you saw today, we have some exciting topics to discuss. We want to make sure that you are fully informed on all the work and all the applications that RNAscope and our wealth of products are engaged in. As you could see from the publication slide that Jyoti showed, we have a number of these webinars in our knowledge resource series. You can see them, watch them on demand on our acdbio.com/recorded webinars page. And if you check out our events page, you can see what's coming up over the next few months as well. So please do check that out.